Welcome back, friends. This will going to be the second video lecture of our complement system. So if you have not watched the first video, then I would like to recommend you please go and watch the first video and then come back here. Then it will be very helpful for you to understand today's video. So first of all, I will going to explain the content of the second video together. So first I am showing you the photos that I have prepared so that you can take a screenshot of this. Okay, so this is the three things that we will going to discuss. So let's start. So in the previous video we have talked that complement system consists of complement components, right? And we are referring them as C1, C2, C3 and so on, right? And we have also told that those complements are synthesized mainly from hepatocytes and then they are present in the normal serum. Okay, now how they activate, this will going to be the main important discussion of today's video. Okay, so they mainly activated by various proteolytic enzymes. Initially they remain in the inactive form, and these inactive forms are known as proenzyme or zymogen. Okay, and when some proteolytic enzyme activates them by cleaving them, they divided into two fragments. Okay, one is the large fragment, that is here this one, and another one is the small fragment. Okay, now although okay, no problem, I am just taking C1 as an example. Okay, so we have learned that proteolytic enzyme will going to activate the complement components, right? And as it is a proteolytic enzyme, that means it have the ability to break the protein. That means they activate complement components by breaking them into two fragments. There is no doubt in that part. Okay, and they usually cleave them into unequal fragment. Okay, one will be larger fragment, another will be smaller fragment. Okay, now those two fragments are usually referred by lowercase letters. Okay, and usually we refer the larger fragments with the help of small b and smaller fragment with the help of small a. That means any complement component, let's say C4, which cleaved by proteolytic enzyme, will have C4b as a larger fragment and C4a as a smaller fragment. Clear? Now the larger fragment will bind with the target near the site of the activation, and the smaller fragment will diffuse from the site and facilitates localized inflammatory response. Meaning, the meaning is C4b will going to take part in further reaction and C4a creates localized inflammatory response. Okay, now here it is there is an important part, that is the each and every complement pathway are a series of reactions. Okay, first if we take the example of one complement pathway, first one component will be activated. Okay, one complement component will be activated. Let's say I am taking again example of C1. Okay, now this activated C1 will activate C2. Okay, now this activated C2 will activate C3, and that is how the entire thing will goes on. Okay, so now let's say we the proteolytic enzyme breaks C4 into C4b and C4a. On the other hand, let's say C2 is also broken by another proteolytic enzyme into C2b and C2a. Okay, now here there is another important concept, that is there is an exception in case of C2. I have just told you that C4b will be recognized as a larger fragment, C4a as a smaller fragment. But due to some historical reasons, the C2a is considered as a larger fragment and C2b as a smaller fragment. And that is only in case of C2. Okay, please remember this. Okay, now these fragments can join together and can form complexes. Okay, for example, let's say C4b2a. Okay, now if that complex is enzymatically active, then we have to draw a bar over them. Okay, so any kind of complement complex that consists of a bar over them, remember that particular complex is enzymatically active. Okay, similarly, if we say C4a2a is enzymatically inactive complex, then we will not draw any kind of bar over it. Okay, so that is the idea. Now, after discussion of that, some normal concepts, now it is important to talk about that there are three important complement pathways. Okay, one is classical, that is the first one to be worked out. Second one is lectin, that is mainly for the carbohydrates, and a alternative pathway. Okay, so in the next video we will going to talk about the classical pathway. Okay, then we will talk about lectin pathway, then we will talk about our alternative pathway. Okay, so for a quick recap, in that video we have learned that complement components together forms the complement system, and this complement component are mainly synthesized from hepatocytes. Okay, and then it will remain in the serum as inactive form. Okay, now they can be activated by proteolytic enzymes, and as it is a proteolytic enzyme, that means it will going to break proteins. That means it is obvious that complement components will be activated by proteolytic enzyme by breaking into two fragments. Okay, and we have also discussed that this breaking is unequal. That means this breaking will break the complement component into two unequal fragments. Okay, and we will denote the larger fragment with the small letter b and smaller fragment with the help of small letter a. But there is an exception in case of C2, right? In case of C2, the larger fragment will be C2a and smaller fragment will be C2b. Okay, so now we have talked about these components can together join to form complex. Okay, and this complex, if enzymatically active, then we have to draw a bar over them, and if they are not active, then we will not draw any kind of bar. Okay, and another important thing, the larger fragment will bind with the target near the site of activation, and smaller fragment will diffuse and facilitate inflammatory response. Okay, so I hope this video will be helpful for you. Thank you.